I'll start by introducing the group that I work with at UC Berkeley, the Redwood Center for Theoretical Neuroscience. And so let's see. I'll just Matt, you're away.
No, I think it should move there.
So it's basically a group of faculty, students, and okay.
Yeah.
Okay, here we go. Okay. Students, postdoc faculty, who basically are coming from a variety of different backgrounds, from math, from physics, from engineering, and we're trying to apply many of the quantitative methods in these fields to understanding the brain. And we're trying to develop computational mathematical models of neural function. So, and as Piero mentioned, we have a weekly seminar series in Wednesday, and you're welcome to join us then to find out more about what we do. So, so the idea here, what we're trying to understand is how neural circuits in the brain process information. The work that I do in my own lab is focused specifically on the visual system. And that's the part of the brain back here in the occipital cortex, the back of your head, that receives information from the eye via a nucleus within the thalamus. Okay, so a lot of what we know from neuroscience is basically based on sort of putting electrodes in the brain and probing what the neurons are doing and trying to sort of explore the system a little pieces at a time and sort of building up a lot of data, trying to figure out what these neurons are doing. And the approach we take on my own lab is more of a top-down approach, trying to sort of formulate theories of what we think might be happening in this system, based on first principles, based on mathematics, basically computational principles, and then working together with experimentalists to try to see if that's actually what is happening. So this is, as you can imagine, a very daunting endeavor, trying to understand the brain. And so before I launch into studying, actually, I'm sorry, okay, went off here. This is just sort of the three different approaches that we take in neuroscience. So one is based on, as I mentioned, sort of like recording from neurons in the brain, trying to figure out what neurons are doing, specific circuit properties and the neuroanatomy of the system. Another approach is just studying the brain from the outside without even putting electrode in the system, just characterizing behavior. And this is basically what psychology does, or psychophysics. And this has been a very powerful method for making very useful inferences about internal mechanisms going on inside without ever actually recording from neurons. And this is sort of the last, sort of the third tier, the area we're working in is trying to formulate theories of what we think might be happening, and then trying to see if those work together with experimentalists to see that that's what happened. That's actually what. Okay, so before sort of launching into study, what I mentioned, like we always talk about the brain, and I think it's important to bear in mind that there are lots of different kinds of brains out there, besides our own, magnificent cerebral cortex. And so and this is kind of really stunning, the degree of sophistication you see in very simple nervous systems whose brains are just a very tiny fraction of our own. And so one example, this is the jumping spider, which has an elaborate visual system consisting of 8 eyes. And as opposed to other spiders that build a web, it doesn't build a web, it relies upon its visual system to hunt and capture prey. And it recognizes conspecifics from vision, although just in the pattern. The sand wasp uses a compound eye. So this animal has very blurry vision as compared to this animal here. The sand wasp has a compound eye and built its nest in the sand and goes hunting for European honeybees. And it hunts for bees as far as a mile away from its nest in the sand, and it finds its way back to its nest by basically following visual landmarks, and it finds its own nest within the sand, basically by memorizing the pattern of the sort of twigs and little sort of debris that happens to surround the nest. Okay, so it can basically do pattern recognition with this very low resolution eye by scanning around the scene. This is sort of a very unlikely system. The jellyfish doesn't even have a brain. Yet it has a very elaborate visual system, so it has a total of 24 eyes, the box jellyfish. And what's really amazing about some of these eyes that has been recently discovered have almost perfect optics. Okay, so if you just take in the lab and probe them optically, they're almost aberration-free. So it has a really highly evolved well-functioning visual system, and it's thought that they use this visual system to navigate within the banks of the river to sort of figure out where they are and where the prey is. So that's just a little bit about the animal world. And one of the things that sort of mystifies me about this, and a lot of my colleagues, is that when we study the human brain, we're often trying to understand how this brain here does things like navigation, pattern recognition, how it computes motion in the world, and all of these things are done by these animals here. Again, with a very, very, just a wisp of the cortex that we're studying. So I think it sort of leaves you with a sense of awe, the sense of mystery that we face in neuroscience. It's really daunting. Okay, so a little bit about digging into the system. What do we know? What do we know about the mammalian visual system? A lot of what we've learned about vision is from studies in higher mammals, especially the macaque monkey. And this is just a picture of the cerebral cortex shown from the side of the macaque monkey. So the front is facing this way. This is where the eye would be. The colored regions are those areas having to do with vision, visual processing. In the macaque monkey, they comprise about 50% of the surface area of the cortex of the macaque monkey. In us, it's maybe about 20 to 25%. Okay, and each of these colored regions is a distinct visual area within the cerebral cortex. And they're organized in sort of a hierarchical arrangement, and this is sort of been learned from neuroanatomy. This is kind of a wiring diagram that's been put together through many decades of neuroanatomy, where, so this is the area V1, this box here, which is the first visual area that gets information from the eye. It basically builds a kind of neural network in space, and it feeds out its output to another area, V2, and then they feed on to a whole complex of other areas which are involved in sort of forming more abstract representations of the visual world. These areas having to do with object recognition. These areas having to do more with motion and spatial navigation. Okay, so sort of digging down to this a little more. So if you take a cross section of this of the cerebral cortex. What we see is that the cerebral cortex is basically a two-dimensional sheet of neurons. It's intrinsically flat. It's basically, think of it as like a sheet of paper. It's 2 millimeters thick, and that's where all the neurons live. And the sheet is just folded in on itself, and that's what you're looking at when you look at the brain. There's all these folds in the sheet. This is taking a cross-section of that sheet, showing the neurons in that 2-millimeter section. And all this stuff here, the so-called white matter, which is just wires. Okay, it's just wires connecting all these neurons together. So there's no computation happening here. This is just sort of wire sending information. All the computation is happening in the purple region here. Okay, so if we zoom in on that a little more, take a little cross section of that. This is what a cross section of the cortex looks like. There's a highly sort of layered structure here, where the input fibers come into layer 4. They project to these layers, and the neurons in the superficial layers, which in turn project back down here. And in a square millimeter of cortex, there's about 100,000 neurons. Just to give you an idea what that means, it's about, that's just what 100,000 looks like as a stadium on a full day. So try to imagine this many neurons packed into a square millimeter surface area of the cerebral cortex. And mind you, this is like all the neurons, like that jumping spider that I showed you that has that very sophisticated visual system. This would be like the entire jumping spider brain, okay? And it's just like 1 square millimeter of the cortex of the area V1. Okay, and then if we could now just sort of any one of those people there is an individual neuron, so we can pluck one of these people out of the crowd and look at it in detail. It's kind of astounding that at this stage of neuroscience that we still don't have a good computational description of what a single neuron does. The simplest model, which is wrong actually, is that it's kind of an elaborate AND gate or OR gate, like in a computer, that it just sort of takes a sum of all the inputs coming in and applies some threshold to the soma. But it could be as complex as entire Pentium chip, and it's probably closer to that end. The more we sort of learn about these neurons and their computational complexity. Zooming in a little bit further, so the inputs come into these. Any given neuron is getting inputs from about 1,000 to 10,000 other neurons. And if I take a cross section of the tissue they're embedded in, this is what you would see. All these sort of wires just coming together. Each one of these sort of round things here we see is the outline of the individual cell, and where they come together, and you see that vesicles bunched up on either side. That's where there's a synapse, where there's signals being sent from one neuron to another. And if we zoom in on that structure, then we see all this fine molecular machinery. This is kind of really sort of the essence of computation. This is where all the signal processing is really happening in these neurons as signals pass from one neuron to another. It's a very complex electrochemical cascade. And there's a lot of computation happening here just in the molecular machinery. Okay, so just kind of bouncing all of that, that's kind of like, the short, like 5-minute tour of neuroscience, and there's a lot that people are learning there in these detailed mechanisms. But what I want to try to touch upon today is maybe 2 ways I think that this is things that are sort of spinning out of this field, and ways we can ways that will affect technology and also potentially art in people, things that people think about here as well. So there's 2 areas I think in technology where I think this is going to be particularly useful as we learn more about the visual system and how the visual system represents information. And one is in the design of video displays, how we format information to be displayed to humans, and how we compress it. Okay, because when we send visual information through the internet, you're not just sending pixels. The image is reformatted into a different code. And ideally, we want to code the image in a way that's similar to the way your brain codes it. Okay? And then the other is in doing image search and recognition. So I'll just talk about these briefly. So this is one example of image coding. How we code color images. So you might think that sending a color image would cost you maybe 3 times as much in terms of like number of bits that you have to send. It might cost you 3 times as much as sending a black and white image, right? Because when you send a black and white image, that's just sort of just one value you're sending at each pixel. And when you send a color image, you're sending 3 separate values, a red, green, and a blue value at each pixel location. But it turns out that sending a color image only costs you about 10% more in terms of bits or bandwidth than sending a black and white image. And this is something that people learned early on in the days of color television. And they're actually, to do this, to send and transmit color images, they're exploiting something that we know about the design of the human visual system. Okay, so this is basically how color signals are coded in television and also in image compression on the internet. Is you start with a color image like this, and you decompose it in these 3 channels, not into R, G, and B, but rather into one channel which encodes the luminance, basically how all the colors are going up and down together. And another channel which kind of codes, well, it's really more like red minus blue, but here it looks like orange minus light blue. And another which codes this, okay, so looks kind of like purple minus green. Okay, this is kind of how they turn out. So basically, in these channels, you're simply coding color differences. Okay, there's no information about luminance here. It's complementary to all the information here, and this has just luminance only and no information about color. Okay, these images here, these color images, are turns out severely blurred and downsampled. So whereas this image here, this luminance image, is presented at very high resolution, very high spatial detail, these images here are blurred and sent at very low resolution. That's why it only costs you about 10% extra bits to send a color image. And this is exactly what our visual system does. So the visual system basically takes a color, the information coming from the cones. It's not RGB, it's basically long, medium, and short wavelength cones in the retina, and kind of reformats it in this way where it has 2 different types of neurons that compute these color differences. They blur over the color information spatially and then downsample it. And then when you then take these blurred color images and add them back together to reconstruct the original, you can get something that basically, to you or I, looks perfect when we look at it. But if you actually pointed a photometer at the screen, you see the colors are all wrong. Okay? The information there is not veridical, it's not correct. Okay? It's not the actual true colors that should be there. But when you or I look at it, it looks just fine. And that's because basically we took the color image, we reformatted it in a way that's similar to the way your brain is formatting it. We threw out the information that your brain's not using and then reconstructed it. Okay? So that's what I would argue we want to do with video information. We're currently not doing that at all with video coding. The way video coding is done today is a kind of very hacky thing that only like a computer scientist would do. Okay? And so what we want to try to do is figure out how to code video information that in a similar way that the visual system basically takes the time-varying image and breaks it down into different spatial and temporal channels, and it subsamples those different channels at different rates. Okay? So if you could figure out how to sort of encode videos in a way that matches that, then potentially we could have higher quality video for much lower cost in terms of storage and bandwidth. Another thing that I think is very exciting on the frontier is trying to exploit higher resolution displays in terms of temporal resolution. So this is just showing us some measurements of what's called the contrast sensitivity function in vision. This is what I mentioned earlier, where psychologists can just make measurements of the system from the outside and infer things about internal mechanisms. And so basically by conventional wisdom, you're blind past about 60 hertz. So that means when a light is flickering faster than 60 hertz or so, you can't see it as flickering. It looks constant to you. And this is why film looks continuous, even though the light bulb is sitting there flashing. Actually, for film, it's like the frame rate is 24 frames a second, but the film is flashed twice per frame. So it's actually flashing about 48 hertz. Okay, so by conventional wisdom, you're sort of blind past this point. But we have reason to believe from a lot of the physiology in the system that your actual what's called flicker fusion frequency is probably way out here, maybe at 200 hertz or beyond. Okay, so this is really the frame rate you have to go to before you actually see something as real, right? If you really sort of wanted to show, just put an image on the screen here, so that you'd be sitting in this room and you'd think you were looking out a window, right? What would it take to fool the brain and make you think that you're looking at reality, right? Because when you're sitting in a movie theater, you don't think you're looking out a window. You think you're sitting in a movie theater and looking at the screen, right? And so basically the question is, how could you fool the brain into thinking that this is actually real? Well, to do that, you actually sort of have to sort of respect all the physiological mechanisms that are going in there inside the system. Okay, so and then the second thing I'll just mention is this problem of image recognition. So right now, when you do a search on Google, it's typically a text search, and you type in some kind of a sentence, and it's just matching the characters in the text to characters it finds in documents. What if you want to search for an image, right? Say, find me this person, or find me, that red Porsche that that I saw the other day on the street, or whatever. Well, Google's not so good at that, at least right now. Okay? But it's about to be a lot better. Okay, so the technology for doing image recognition is dramatically improving. And it's improving in part because they're basically inspired by insights about these computations going in the brain. So to give you an idea why image recognition is so difficult for computers, this is just showing you two different images of the same scene, but we're showing you the information of the image in a way that you're not used to seeing. Here we're just plotting each pixel intensity. This is just black and white image. Each pixel intensity is the elevation. Okay, and this is the spatial dimension, the horizontal spatial dimension, and the vertical spatial dimension. So we're just plotting the image intensity as a function of space, as a function here. So these are two different images of the same scene. Any guesses of what the scene is here? Okay, so that's what it is. That's what we're looking at in each case. This is the image of that scene there, and this is the image of that scene there. Okay, so when we look at these images, it's effortless for us to see those are basically the same scene. We can see the bicycle and so forth in the foreground. And now when you see actually the raw data here, you can see why it's going to be so challenging for a computer, because look at all this light here in the background where the light is shining through the trees. It's creating all these strong variations in the pixel intensities here, right? So the computer is saying, oh wow, this is really important stuff, right? And where the bicycle wheel is here, well here you can kind of see it now, but where the bicycle wheel is here, it barely shows up in the pixels. Okay, so these are all things that our brain is doing when we look at an image. There's those massive amounts of neurons you saw in that V1 and V2 in that map, right? A lot of it is just trying to extract features, sort of group things together, and get together a story about this scene. So this is sort of what all this machinery is doing. So one of the things that people in computer science have done now is sort of inspired by this architecture, seeing that the visual system doesn't just solve this problem, just doesn't just sort of take the bicycle and put it through like a program and then out comes the label bicycle. It basically tries to take the image and break it into a set of features and does this in a kind of piecewise process in a series of processing stages. And so people have been developing these so-called deep networks or deep learning algorithms. You might have read articles about in the press fairly recently, where you start with the image here and basically try to extract features from the image at different locations and different positions locally. And you do that again, and you sort of form a feature map here. This is kind of like that area V1. and you do it again, you extract the features on this part here that V1 is doing, you feed it to another area here, you call this V2 in that map. And you do it again, and you do it again, and you do it again. And you sort of stack these layers. And almost miraculously, well, I shouldn't say miraculously, but it's really kind of astonishing that this output layer here now gives you very high level and very semantically meaningful representations of images, okay, in ways that we weren't able to do before. So just to give you an idea, okay, so yeah, I'll just wrap up here. This is maybe just the last one. The performance of some of these systems. So if you handed an image like this, the correct answer is container ship, and indeed it labels as a container ship. Here, for example, the correct answer is mushroom. It actually labeled as something else, but mushroom was the second category here. But I think if you asked me 10 years ago, would this be possible for a computer to achieve this level of performance?
Yes.
Can you say a few words about your book?
So I was a co-author on that book. My friend Kyle, who manages a translational medicine master's degree at UC Berkeley, he was in his PhD program at the time, came to me with the title, and he said, we should write this because I've got a really good title. And I said, that is a good title. That's the reason why his name comes first on the book, is because he titled it. It's basically an exploration of biotechnology in video games and movies and television. And we'll sort of give examples from popular culture and talk about the science that we think inspired them. Sometimes we'll say when they're way off, and sometimes we'll point out when they really did an excellent job of sort of extrapolating from the facts. Available on Amazon.
Yes, question.
What's phage? One of the terms you used was pH.
Oh, phage.
Phage, sorry.
Phage is a virus, but it's a virus that targets bacteria. Yeah. And there are, underneath the world that we live in, there is a tremendous diversity of bacteria and a tremendous diversity of phage trying to take advantage of the fact that there's all those bacteria. And phage are sometimes used just as tools in biotechnology because you can use them to manipulate bacteria. But if you were to quantify the number of phage in this room, I'm guessing it would be significant.
Can you just clarify the distinction between a molecular biologist and a synthetic biologist?
No, I can't. So molecular biology, there was a paper that came out a couple of weeks ago where a molecular biologist was making fun of the lingo that synthetic biology has come up with, that there's already lingo in molecular biology for. But anytime there's, especially in academia, but anytime there's a new kid on the block, like we're not going to call ourselves molecular biologists. You're the old fuddies. We're going to call ourselves synthetic biologists. We're going to invent our own language because then you have to be in the club to understand what we're talking about. Also, sometimes because we're using fundamentally different viewpoints to look at things and construct things. So I don't think there is a hard and fast rule. I would say instead, depending on who you talk to and what conferences you go to and what techniques maybe you use, you will either self-identify as a molecular biologist or a synthetic biologist. I will work with anybody who can do stuff that I need.
From your talk, it seems like it's about end product in this way, like what your end goal is, like you can manufacture. I can't quite tell. I'm always confused about this.
Well, that
suddenly becomes synthetic biology, but it seems so.
Yeah, that might be an engineering bias too, because I tend to frame things as building things as solutions for problems, but there are also synthetic biologists that are based mostly on building tools to manipulate cells on a sort of higher level with a better degree of control. So I guess the answer to that question is I don't know. Yeah. And it's one of those, somebody will write a PhD in culture of science.
Yeah, they will.
And it'll be based on this. It'll happen in about 5 years.
Okay, thank you very much.
Thank you.